And it's a real honor to have today Tonney Nyirenda from Kamuzu University of Health and Sciences to give the first talk of the session. And Tonney, the stage is yours.
All right, thanks, Lyra. Yeah, so this afternoon, evening, I'll be making a presentation on how does malaria parasites compromise immune responses induced by viral or bacterial pathogens, but also vaccines. So I'm Tonney Nyirenda, Associate Professor of Immunology at the Kamuzu University of Health Sciences in Malawi, Africa. So thanks for the opportunity to present. And I'll be presenting on the topic, how does malaria parasites compromise immune responses induced by viral or bacterial vaccines or pathogens? So as you may be aware, malaria is commonly associated with bacterial stream infections, and in particular, non-typhoid or salmonella. And you would find that there are a lot of publications that have really shown that slight positivity of malaria tends to be associated with, in positivity of bloodstream culture of non-typhoid or salmonella. So then, considering our interest, this forms, a good model for us to really study how does malaria, compromise immunity to bacterial pathogens. So just in case you may not be familiar with the interaction of malaria and invasive non-typhoid or salmonella. So in terms of diagnosis, so in the field, particularly in areas where malaria is endemic, it is easy to diagnose malaria because we've got microscopy, but also you've got rapid tests, so short turnaround time. While for bloodstream infections, including non-typhoid or salmonella, you need blood cultures, which will take like 72 hours or more. In terms of clinical diagnosis, it tends to be a challenge for the clinicians for the reasons that you tend to have an overlap in terms of the clinical presentation of malaria, pneumonia, but also other bacterial bloodstream infections, including non-typhoid or salmonella. Treatment, then, because diagnosis is easier for malaria and it's more common as well, then if a child presents to hospital, they can easily be put on treatment for malaria, while bloodstream infections may be ignored, only for the child to come later on with a more severe disease. So when you consider case fatality rates for malaria alone, it's about 10%. But if you have a co-infection of malaria and invasive non-typhoid or salmonella, the case fatality rates increases to up to about 24% or so. So more about invasive non-typhoid or salmonella disease. So it's a salmonella disease, not caused by typhoid, but the other serotypes of salmonella, including typhimurium and enteritidis. We are not interested with gut localized infections. We're particularly interested with invasive disease. So bloodstream infections, or if it also goes into the cerebrospinal fluid as well. So that's what we are actually interested in. So it's an important problem, and there are reports with regard to antimicrobial resistance, but also there are a number of host risk factors that we have to be mindful, including age, malaria, which we're going to discuss, but also things like nutrition, anemia, and HIV. HIV, particularly in adults, but in children, I think the ones that tend to be very common is young age, malaria, current or recent malaria, nutrition, but also anemia. So in terms of the clinical features, so it will present, majority of them with, fever, but also it will present with pneumonia, diarrhea, splenomegaly, but also hepatomegaly. So the blood test, if they are done, usually it's associated with a severe anemia, and this severe anemia could also be in the context of malaria as well. So from the onset, I want to bring to your attention some of the early studies that we had done, because we were interested to make progress in as far as vaccines for non-typhoid or salmonella were concerned. So we wanted to see to it that we can characterize the immune responses in the context of adaptive immunity. So our first questions were, how does adaptive immunity against non-typhoidal salmonella develop in healthy children? So with that question in mind, we studied the acquisition of humoral as well as cell-mediated immunity against Salmonella typhimurium, which is the principal non-typhoidal Salmonella salivary. And we studied this in under 5 children. So we conducted a cross-sectional study to study the development of specific T cell as well as antibody-mediated immunity in these children in Blantyre, Malawi. So we recruited 90 children and 20 adults. So these children were age stratified as shown on the figure, and we performed immunological investigations, including ELISAs for antibody specific antibody titers, serum bactericidal assays, SPAs, T-cell immunophenotyping, looking at the phenotype in terms of the memory, but also looked at cytokine production. So first, let's appreciate the epidemiology in terms of the burden of invasive NTS. So what I'm projecting here is a figure that we generated from the data that we collected from Queen Elizabeth Central Hospital, showing the burden of invasive NTS in under 5 children. Importantly, what you see is that it's common, particularly in young children, and the peak in terms of disease is around 13 months or so. So this is around the time that the child is still developing his or her, antibodies, but also to a certain extent T-cell mediated immunity. Then we looked at the serum bactericidal activity. For the serum bactericidal activity, basically, this is we did ex vivo experiments where we collected serum from children, and we took also an isolate of Salmonella typhimurium and mixed these, incubated for some time, and then did mouse measles, which is a traditional method to do the colony counts to determine whether there's been killing or not. So the killing is supposed to be mediated by the antibody. If a child has got specific antibody, we'll be able to fix complement, and it is through complement-mediated killing that the Salmonella would actually be killed. So on figure A, what I'm actually showing you is the output from those from the serum bactericidal assays that we had actually done. So we see that on the horizontal axis is age, where on the vertical axis is the load in terms of the bacteria. So the figure has been flipped. So the negative is on top, the positive is down on the vertical axis for easy for you to really to be just so that it is easy for me to actually explain at this stage. But essentially, a cutoff of -1 and below tells us that that particular child or this sealer is immune, okay? So what you actually see in this particular figure is that you see a dip at around 8 months or so, and then the peak at around 35 months of age. So between 0 and about 8 months, there is a decline. So this decline is expected because the child is losing maternal antibodies, and at around 8 months, that's when you actually have got a dip. And then gradually that particular child will be gaining antibodies that he or she has developed on his own. And that is what actually explains the serum bactericidal activity increase with age in this regard. And then when you consider the CD4 T cell responses, so essentially we had these intracellular cytokine staining experiments that we had actually done, where whole blood was actually stimulated with non-typhoidal salmonella, particularly Salmonella typhimurium. And then we looked at a number of cytokines. So what you're actually seeing here is a percentage of the total cytokines that we had measured, including interferon gamma, IL-2, and TNF-alpha. For this, we see that the antibody, the cytokines can actually be detected, from early age, peaking around 14 months or so. It does decline, but doesn't decline completely, as you can actually see. Now, we attempted to superimpose the kinetics in terms of the antibody-mediated immunity, but also the T-cell-mediated immunity with the cases that I'd shown you earlier. So what you actually see is that at the time that what you have as spikes in black are the cases. What you have in lead is the CD4 cytokine production, and what you see in dashed lines, this is the serum bactericidal activity mediated by the antibody. And what you see is that at the time that you have got a dip in the serum bactericidal activity, that's when you have got a peak in disease. But also that's the time that you've got a peak in the CD4 cytokine production. So what does this really tell us? So basically, you are seeing an increase in serum bactericidal activity associated with the decline in incidence of Salmonella typhimurium infections or invasive Salmonella typhimurium infections, suggesting that serum bactericidal activity controls invasive, Salmonella typhimurium in older children, because what we are seeing is that, as the serum bactericidal activity is increasing, from 18 months or so, that's when you actually see the decline in cases of invasive NTAs. So this would suggest that antibody-based vaccines would be required to prevent invasive Salmonella disease, particularly in susceptible children. Again, just to remind you that we had considered, we know of a number of host risk factors in as far as invasive NTS disease is concerned. So our next, having characterized the acquisition of adaptive immunity in the context of antibodies as well as the T cells, we then moved on to do more studies with regard to malaria. So we had a number of questions in mind. How does malaria parasites compromise humoral as well as cell-mediated immunity in children? Does the serum bactericidal activity in NTS vary with age or antibody titer during malaria? What explains the loss of serum bactericidal activity to non-typhoidal salmonella during malaria? So we conducted a longitudinal study, and this longitudinal study, we had children presenting with uncomplicated malaria, children presenting with non-malarial febrile illness, but also healthy children. And these children were from Blantyre. And we had considered, to age stratify the children because we know that, stratifying age is important because immunity against malaria, against, non-typhoidal salmonella is age dependent. So we considered all age groups. So we had a group of children, 50 of them, and uncomplicated malaria, 15 non-malarial fever, and the healthy children. And then we followed up we recruited them at day 0, and then followed them up at day 14 or 2 weeks down the line and 1 month, down the line. And we did more or less similar experiments in as far as the serum bactericidal activity is concerned, but also measuring the antibody titers. But also we did some, flow cytometer-based complement deposition experiments, but also respiratory-based. And this time around, we had considered to start from 6 months of age for the reason that we knew earlier on that, between month 0 or from day 0 to 8 months or so, the child is really, immunity is actually coming from the mother. So we didn't want any confounders. So we opted to start from 6 months when you actually have got a dip in terms of serum bactericidal activity against non-typhoidal salmonella. So when we did the serum bactericidal activities as experiments, this is what we had actually found. So on this particular figure, the figure is not flipped. So again, you actually notice that the negative is down and then the positives are actually up. So for the cutoff is also, at -1 log change in serum typhimurium. I mean, log change in Salmonella typhimurium colony-forming units per mil. So what you actually see is that during acute malaria, a good number of children are not killing well. That's why you have got a median of, greater than around 0 or so, which is actually above the threshold that would consider protection. And then also at 2 weeks down the line in convalescence, there is still some children that have not fully recovered. Later on at a month, so these are the same children that are being followed. The killing is not, is at least at this, the median is similar to the level of those that are we considered as controls in this case. The controls, non-malarial fever, and the controls with regard to malaria, okay? So, so what we had here, non-malarial fever is the healthy ones, and then the non-malarial fever, and then you have got, those ones that had no fever at all, so they are just healthy ones. And these ones were, to a certain extent, in terms of the medium, comparable to 1 month down the line. So we assessed, what we had seen in the context of age, and we found that if you consider C, you see that at 18 months or so, you have got, going onwards, you've got a good number of those that were not able to kill, and I've highlighted these in the box, which is a dashed box. So you see a good number of them that are not killing well. So there's poor killing that you actually see. In B is non-malarial fever. For those that are older than 18 months of age, they are generally killing very well. And this is actually comparable to those that were healthy. So then we probed further those that had got malaria. And indeed, as pointed out earlier, those that were older than 18 months, but had malaria at acute stage, the killing, a good number of them, they were not killing very well. So up to 62% had poor killing. 2 weeks down the line, 56% had also poor killing. 1 month down the line, 35% poor killing. So to a certain extent, telling us that the impact, much as it appears to be transient when you have got malaria, that there is this poor killing, but it also lasts for quite some time. So here we're looking at the space of 30 days or so. So further to that, we assessed this in now, not just with age, but in the context of antibody titers. And what you see on this particular figure, particularly during acute malaria, is that there is loss of killing even in children that had high antibody titers against Salmonella typhimurium LPS. So as you can actually see where I've actually highlighted in the cycle there. Then we went on to assess, because we know that complement, you can assess complement using considering different components of complement, and the killing is mediated by complement. So we assessed the deposition, C3 deposition, but also we assessed, the terminal events in terms of MAC deposition, so C5b to 9. And what you actually see is that during acute phase, the deposition of C3b, which is a component responsible for opsonization, is reduced during the acute phase and does increase. The deposition does increase a bit at 2 weeks, more or less remaining similar to 2 weeks at a month or so. Where in the case of controls, healthy ones, non-febrile, and those that were febrile had fever, those ones, the deposition was actually higher compared to all the malaria groups that we had actually considered. So similar, not necessarily a similar trend was in the MAC, but only at the acute during malaria, that's when you had significant reduction of the MAC or C5b to 9 in terms of deposition. So to a certain extent, this reduced complement components deposition may explain the loss in the serum bactericidal activity that we had actually observed. Furthermore, we looked in the context of the cells. So we did the serum bactericidal, we did the respiratory-based experiments on the flow cytometer, and what we found is that there is also reduction in this respiratory-based, more prominent, particularly during the acute phase of malaria, and the respiratory-based peaked a bit at 2 weeks and a month down the line. And when you compare to the controls, the non-malarial fever also was impacting on the respiratory based, but not the healthy ones. In this case, the respiratory based was actually more robust. So considering the investigations that we had done and what I've actually shown, we can say that we're saying that we say that there's loss of serum bactericide activity during malaria, and the loss in serum bactericidal activity during malaria is also seen even in older children, even those children with high specific IgG titers. We see reduced complement deposition. This may explain the loss in serum bactericidal activity. There was defective serum bactericidal activity during malaria in children with high antibody titer. This may suggest that malaria may compromise even vaccine induced responses, because in the field you'd give the vaccine and hoping that once the child has got the antibody, then they are good. But with malaria, then even if you would have high antibody titers, that immunity tends to be compromised. I think the comfort is that the loss in this immunity tends to be transient, as we had actually seen that, there seems to be an improvement 30 days down the line. So we are still conducting more studies to understand this further. So we've got 2 PhD students that are doing, system serology. So we've got Agnes Lakuzala, who is doing, her PhD is on system serology, now particularly looking at Salmonella antibodies in children presenting with uncomplicated malaria, but also varying ages. And we have got Josephine Banda, who's also doing systems serology project on malaria parasite antibodies, looking at children with asymptomatic malaria and complicated malaria, but also cerebral malaria. And we look forward to their results. They've started already generating results, powerful results that will help to explain most of the things that I have not explained this time around. We also asked, does malaria parasite compromise vaccine-induced antibody effector function? And for this, this is coming from the preliminary results that we've just looked at. So we did, a cohort study of COVID vaccine. So we, participants were given the JJ COVID vaccine. So we had healthy adults, HIV-infected adults, but also adults who had just recovered from, malaria. And these adults were given the single dose, and they were sampled at baseline, 4 months down the line and 5 months down the line. And we did investigations looking at antibody titers, but also looking at the neutralization using the surrogate neutralizing assays. So from this study, what we actually see from figure A is that in children, in adults with malaria, at day 0, of course, at day 0, they had recovered from malaria. So it's a malaria group, HIV group, and the HIV group. Not that they had malaria this time around. But we will see that immunization with COVID vaccine actually induced antibody titers specific for COVID. So this is S1 IgG titers. For HIV as well, it does induce, much as we didn't find any significant differences, but you could clearly tell that 4 months down the line and 5 months down the line, there was an increase. In healthy individuals, that's where we had found a significant difference compared to day 0. And then we assessed the neutralization. And when we looked at the neutralization, we were surprised to see that the neutralization was actually didn't change in the malaria group. Compared to the other groups that we had actually examined, the HIV infected group, but also the healthy group. And considering what we have been observing with respect to non-typhoidal salmonella, we think that maybe there should be some immune defects that are actually induced by malaria in this particular group. So JG COVID vaccine, it does induce increase in antibody titers in all the groups. Vaccination induced increase in the neutralization titers in the healthy as well as HIV infected individuals, but not in the malaria group. We observed that the malaria history was associated with poor correlation in antibody titers and neutralization titers in this case. So in conclusion, malaria compromises cellular as well as humoral responses to viral as well as bacterial pathogens and vaccines. Mechanisms of these malaria-induced immune defects are not fully understood, Systems serology studies will help to explain malaria-induced immune defects, but also systems serology studies will help to determine the protective immunity. There is a need to optimize immunological interventions in the malaria endemic settings, so that some of these things that we have just highlighted can actually be, addressed. So thanks to the people that were involved, and also the funders. But just to highlight that we have the microbes and immunity vaccines group, which I lead. Our interest is blood in the context of the bugs, as well as in the immunity, antibody-based immunity against infectious diseases, but also non-communicable diseases, but also vaccine immune responses are areas of our interest. And we've got platforms that we can do fantastic immunological studies. Thank you for your attention. Thank you.
Thank you, Tonney. Excellent and indeed very powerful data to share with us. So thank you for sharing that. Because we had some technical issues at the beginning, we are going a little over time, and I don't want to eat into the next speaker too much. There is one question already in the chat, so I'm going to suggest we answer that question, and then we can move to Shikha. Should I read the question, Andrea, or do
Yes,
we let
I can't
Patrick?
see the question. Yeah.
So this is from Patrick So. Given that malaria might compromise vaccine-induced immune responses, what are the implications for the efficacy of current vaccination program for measles, tetanus, or other common childhood vaccines in malaria endemic regions?
Yeah, thanks for that question. I think it's an important question. So this is the thing that we've been struggling to answer, and that's why in conclusion of my presentation, I say that we needed to see to it that perhaps immunological interventions are optimized. So there are some things that I think are done in the field, maybe without necessarily appreciating the biology in terms of what is actually happening. So like, for instance, if somebody has got fever or has got malaria, it's not the best time to actually give the vaccine. So you would want to clear the malaria first, and then you can take into account the vaccination at a later stage. But otherwise, over and above that, then we have to be mindful that malaria has the potential to compromise vaccine-induced responses. Are there things that we can actually do? Can we optimize our the antibodies that are induced to see to it that even with malaria, we can actually be able to deal with that? So that's the big question that even us, we want to understand and be able to, optimize where we can. And that's why we are actually doing the systems serology, so that then we can be able to have a better answer in terms of how we can move things forward. But this applies not only to the vaccines or the bacteria that I've actually talked about, but I think it should be a general thing, and should be a concern to us, and we have to do something about it. Thank you.
Thank you very much, Tonney. And
Thank you.
now, oh, I see that Stephen has a question. Stephen, if you can maybe write it in the chat, and Tonney can answer you, because we only have 20 minutes left for the next speaker. Thank you. Sorry to cut the discussion short, but the discussion can continue on the chat.
Oh, thanks.
And I will invite our next speaker, which I won't present in as many details as I wanted to present, again, just to keep to time. So Shikha from Glasgow, who's going to speak to us about RNA biology in mitochondria in apicomplexan. Shikha, please go ahead.
I'm trying to share my video as well. Okay, cool. Can you hear me?
Yes, hear you and see the screen.
Cool. Thank you very much to the organizers to give me the opportunity to talk about my work. I must warn people that I have a little bit of a cough, so if I start coughing in between presentation, I apologize if there's a huge disruption. So hello, I am Shikha. I'm a postdoctoral researcher at the University of Glasgow. And today I'll be telling you a little bit about my postdoc work, which involves a beast of a machine called Toxoplasma mycorrhizome, and the incredible discoveries that we've made on it in the lab. So starting with a little bit of a background, Toxoplasma gondii is a single-cell intracellular parasite that belongs to the apicomplexan phylum, which consists major parasitic protists, including Plasmodium that causes the deadly malaria, as you heard Tony talk about it in the previous presentation. And Toxoplasma is often deemed as the most successful parasite, as it can infect virtually all warm-blooded animals, and with global infection rates reaching up to 30%. This parasite can cross the blood-brain barrier and forms dormant cysts inside your brain that can stay there forever. And although asymptomatic in healthy people, infection in immunocompromised individuals or pregnant women can have severe consequences. And if that doesn't convince you, the world's richest man also thinks that it's a danger to our democracy. So overall, an important pathogen to work on. So apart from being medically important pathogen, it's also Toxoplasma has also become a versatile model organism. And the more we study all of these, the different branches of eukaryotic tree, the more we find out that the fundamental principles of biology that we thought are universal because they are true for humans and yeast, we find that they are actually quite divergent among the eukaryotes. And one of the best example of this is the Toxoplasma, the single Toxoplasma mitochondrion, which exhibits unique features. For example, it has an extremely reduced genome of just 6 kb that contains only 3 protein coding genes. And more importantly, at least for this talk, has extremely fragmented ribosomal RNA. So we know from our textbooks that usually 2 ribosomal RNA makes a ribosome, and it's important for protein synthesis. In Toxoplasma mitochondrion, these rRNAs are in fact fragmented on the genome into tens of pieces. And previously we found more than 30 pieces based on the homology with the bacterial ancestor. However, um, with such an extreme fragmentation, we don't know how this mitoribosome assembles, how it looks, and more importantly, how it manages to translate the 3 protein coding genes that are in the mitochondrial genome, which are important for parasite survival. So the questions that we asked was, what is the composition and structure of the Toxoplasma mycorrhizome? Is it reduced, or is it even bigger than the ancestor, than its ancestors? What is the true extent of RNA fragmentation and reduction? So remember, the information that we had was based on homology with the bacterial ancestor. We don't really know the complete picture in Toxoplasma mitochondrion. And then finally, we know that there is extreme fragmentation. How does the mitoribosome assembles despite that? So to answer our question in the most efficient way possible, we turn to single particle cryo-EM, and we worked with collaborators, Alex and Victor. Alex is a PI at Helsinki, and Victor is an incoming PI at Glasgow. So we ended up tagging a known mitoribosomal protein and pulled down the native complex to reveal and resolve the cryo-EM structure of the mitoribosome, which is a massive 4.3 megadalton complex containing 124 proteins, and more incredibly, a massive 53 rRNA fragments, so more than what we knew before. And this RNA fragments are, of course, overlaid with the known conserved mitoribosomal proteins. And then we found 55 clade-specific proteins shown here in color. And these are overlaid or on top of the conserved mitoribosomal proteins, indicating that these proteins have been acquired and fixed throughout the parasite evolution in the mitoribosome. So as I said, we found an incredible 53 RNA fragments that make up this ribosome. And this really marks a paradigm shift in ribosome biology, where we know from other systems that there are only two long RNA molecules that make up the ribosome. And these RNAs already contain intrinsic information that helps the RNAs adopt secondary structures. And the assembly of the mitoribosome is then co-transcriptional as the RNA is transcribed. However, in case of toxomitoribosome, most of these RNAs are just too small, around 40 nucleotides, and they cannot self-fold, meaning that they need factors in trans that can actually help these RNAs to fold and assemble into the mitoribosome. Incredibly, we also found many of these RNA sequences in the ribosome actually share part of their sequences with each other. So two different RNAs have some portions of their sequences that are exactly the same. And not only are these sequences or RNAs found with same sequences found at different locations in the ribosome, they are also, same sequences also folded differently based depending on which part of the ribosome they are in, emphasizing the fact that this folding is really dependent on the context of the ribosome or the interactions that are taking place between the ribosomal RNA and the proteins. So when we mapped these ribosomal RNA on the genome, we thought, okay, different RNAs come from different genes. However, these RNAs containing similar sequences are actually originating from the same part of the genome, which is something that is not known in any other organism. And in total, we found 9 of these reused sequences in the mitochondrial genome. And this suggests that there must be, among other things, there must be complex factors at play that is involved in actually processing these RNAs to their final mature form, which we hope to uncover in future. So until now, I told you about the incredible RNA features of Toxoplasma mitoribosome, and we know that there is extreme fragmentation, there is reduction, but now I'll present some of the strategies that the parasite adopts to actually circumvent this challenge of extreme RNA fragmentation. So one of the things that we found was that most of the RNAs in the mitoribosome are actually polyadenylated at their 3' ends, as you can see the red, as you can see in red here. And these, we think that these poly-A tails of the rRNAs are actually involved in basically stabilizing the RNA and helping it interact with the proteins in the ribosome. And we also see that these rRNA poly-A tails are actually interacting with several proteins in the ribosome itself. So we hypothesized that these poly-A tails basically act as handles for proteins in the complex that can that can help bring the ribosome together. And so we knocked down a mitochondrial poly-A polymerase to test our hypothesis. As you can see here, this poly-A polymerase is essential for parasite growth. And when we looked at the RNA, we looked at the poly-A length of RNAs through RNA-seq. We saw that many of the ribosomal RNA now manifested shorter poly-A tails. And when you look at the mitoribosome assembly itself, we find that the mitoribosome cannot assemble anymore, even though the proteins that are needed to form the mitoribosome are present, telling us that this is a specific assembly or a stability defect for the mitoribosome. Another strategy that toxomitoribosome adopts is the presence of numerous helical repeat proteins. We know from many other organisms that helical repeat proteins are RNA binders. What we saw in our structure was that these helical repeat proteins, marked here in purple, are in fact helping different rRNAs co-localize with each other. So kind of bringing them, binding one protein, binding a few RNAs and kind of bringing them together. And some of these helical repeat proteins, interestingly, belong to a specific family of RNA binding proteins called RNA binding domain abundant in apicomplexa, or RAP. And as a specific example here, I'm showing that one of this RAP protein called ML138 is actually binding 6 different rRNAs together. And we think that it's basically involved in bringing all these fragmented rRNAs together into the mitoribosome. Of course, when we knock down this protein, we see specifically that the mitoribosome cannot form anymore. So at the end of my presentation, I would also like to draw your attention to a specific family of protein called AP2 proteins, which are quite well known in the parasitology or apicomplexan biology field. We know that the AP2 proteins are involved are act as transcription factors and involved in basically binding to DNA, and important for life cycle stage switching, specifically in Plasmodium. A lot of work has been done. We found that in our structure in toxomitoribosome, there are 4 AP2 proteins that are part of the complex. So this is the first time that somebody has shown that the AP2 proteins are not just DNA binding, but also can be RNA binding and potentially involved in RNA regulation in these parasites. And these proteins are not just present in the mitoribosome, they seem to be involved in specific functions and compensating for the extreme RNA fragmentation and reduction in the complex. As an example, if you look at the protein in green, which is an AP2 protein, it is present at the interface between the large and small subunit. And if you look closely, the N terminal of this protein is buried in the large subunit, whereas the C terminal of this protein is extended into the small subunit. And this C terminal actually runs parallel to an RNA molecule that is present in other organisms, but is extremely reduced in toxoplasma. And this RNA called H44 is actually involved in maintaining this inter-subunit associations, which is important for the ribosome function in general. And we see that this function of the RNA has now been taken over by this AP2 protein. So at the end of my presentation, I would like to summarize by saying that, I highlighted all the remarkable RNA features that the toxoplasma mitoribosome exhibits. And this in a way, Toxoplasma is providing a magnifying glass to look at ribosome evolution and really what is possible for the ribosome biology itself. And we need more studies to reveal more of these remarkable features that are contained in the parasites. So I would like to thank at the end all the people that are involved in the study, collaborators Alex, Victor, Dario, of course all the people from Toxolab in Glasgow. I work in Leela Schreiner's lab, and it's incredible working with all the amazing parasitologists at Glasgow. Glasgow is an incredible place to study parasitology. So I encourage if any of you are thinking about doing that in future. And yes, thank you. I'll take any questions.
Thank you, Shikha. Do we have questions from the audience already? Let me try to see the audience. Stefan?
Thanks. Nice work. Unfortunately, it's a bit far from my expertise, but I have a very naive question for you, which is concerning antibiotics. Are there any antibiotics that are active against these mitochondrial ribosomes? Because, of course, the ones that we know are toxic in us and therefore being screened out. So I wonder whether you have these available in your labs and you use them. Thanks.
That is actually a very good question, and obviously something that we've also wanted to we also want to look at as well. We know that we already knew that many of these antibiotics actually do not work specifically for AP complexin mitoribosomes because they are so different from their bacterial ancestors, right? But we do know that there are some antibiotics that do work. For example, thiostreptin. There are some reports that it actually works for the mitoribosome. There are some reports that doxycycline also has effect on mitochondrial ribosomes, even though the concentration needed is different from what we know for other ribosomes. But yeah, there are very few that we know that actually work for mitoribosome. And this is something that we all obviously need to look into more because ribosomes are really good targets. And this is something that we plan to look in future.
Thanks.
Hi, Shikha. This is Rahul.
Hello.
Nice to meet you on this webinar. I have one question. Like when you do the conditional knockout wrap or the ML138, how long you wait to see the disassembly of mitoribosome complex? And this disassembly is only for LSE or it's the whole complex is disappear?
Yeah, this is a very good question. So specifically for wrap proteins, I think we start to see an obvious effect on the mitoribosome using native page at 48 hours or so. Of course, this also depends on cell line to cell line. Sometimes the knockdown is very good, and so you see a very early defect. So it's difficult to say exactly where in the pathway the protein the assembly has been disrupted. But we do know that it does disrupt the assembly, and we see it around 48 hours or so. We don't know if so we know that we see the mitoribosome not assembling. We haven't looked at individual proteins by themselves. So that is something that we're also looking at. But we know that at least the complex itself cannot form anymore.
Thank you so much.
Thank you for the questions. I'm sorry that I'm going to stop this at 2 questions because I know we're already over time. Thank you both of the speakers for very interesting and exciting talks. And thank you for those who participated. And those who participated, especially those of you from our African partners, but everybody, please encourage other colleagues to join these seminars in the future. They are free and they're an opportunity for you to enhance your knowledge of the field and to enhance your contacts by asking questions. And you know, the more you do, the more people will remember who you are next time you write a grant or a letter of interest in a postdoc opportunity. So please encourage everybody to join in. Andrea, did you want to say something else before we close?
No, please stay also tuned into our social networks. That is where we usually post the updates on the upcoming speakers. We have next session on the 2nd of December with guest speakers from University of Glasgow as well. And we're looking forward to see you in a month.
Excellent. Thank you, everybody.
Thank you, everyone, for joining.